Today's topic, we're focusing on allergy research in the Adelaide Biomedical City Precinct. So we know allergy has become more common in our society for a variety of known and unknown reasons. The presenters today will be doing a couple of presentations which focus on life-threatening allergies, both taking different approaches from the clinical research approach with patient-based research and the basic mechanistic research using novel approaches. Firstly, I'd like to introduce then Dr. Preethi Elid of University of SA, the Clinical and Health Sciences Division, a Senior Research Fellow. Preethi, would you like to start speaking?
Thank you, William, for the introduction, and thank you to all the organizers as well for the opportunity. It is a pleasure to discuss our efforts into developing a peanut allergy vaccine during this week of World Allergy Week. Just to get us started, the global prevalence of food allergy and the related anaphylactic reactions has increased over the past decade. In Australia, food allergy affects about 10% of the infants and about 2% of the adults. The most common foods which are associated with about 90% of the food allergy reactions is listed in the box here, of which peanut is one of them. Now, peanut allergy has a very high prevalence in Australia. About 3% of the children between the ages of 1 to 14 are diagnosed with peanut allergy, and of whom about 80% of them remain allergic for life. With this increasing incidence of peanut allergy, it is also an increasing awareness of the symptoms associated with food allergy and management skills, starting right from daycare to schools, along with age-appropriate education of children, as well as better labeling of the food products to warn of allergens. Now, until recently, there was no clinically approved product or therapy for peanut allergy. In early 2020, one of the oral therapeutic called Palforzia was approved by FDA, and not as a cure, but mainly as to reduce the risk of an anaphylactic reaction during accidental ingestion. So the patients at all times during and after treatment are still advised to avoid peanuts as well as carry an EpiPen with this. So there still remains, even though there is one approved product, there's still a dire need for a therapeutic that can be administered in a very short treatment plan with little or no adverse events, and which has the capacity to induce long-lasting tolerance. Now, to develop such a therapeutic, it is critical for us to understand what are the key players in the immune response to peanut allergy. Now, in predisposed individuals or upon first exposure to peanut, either through the gut mucosa or through the skin, the T cells at those sites get primed, and they develop a Th2 phenotype, which means that they secrete cytokines like IL-4, IL-5, and IL-13 with very small amounts of interferon gamma. Now, this primed T cells now have a crosstalk with the B cells, and this induces the production of peanut-specific IgE cells. Now, IgE antibody. Now, this IgE antibody gets loaded onto the mast cells, and upon a secondary exposure or re-exposure to peanut now, the cross-linking of the IgE and the receptors results in the release of these mediators such as histamine and leukotriene, platelet activating factors, and these are responsible for the allergy cascade and the symptoms which we now know as as allergic reactions, or sometimes in severe cases as anaphylactic reaction. Now, most of the other other immunotherapies in clinical trials focus basically on either suppressing this T cell responses, this Th2 response, or either by administering blocking antibodies that prevent the binding of the allergen to the mast cells, or in other words, directing the T cell response away from the Th2 cascade, from the Th2 immune responses. Now, in allergic individuals, it is important to understand that this Th2 skewing is very specific to the allergen. For example, on the screen now is a patient who is allergic to peanut, but is tolerant to eggs or albumin. And you can see that it's only the peanut-specific T cells which generate IL-4 or have a higher proportion of IL-4, which in turn translates to a lower Th1, Th2 ratio. Whereas in the egg, this person is tolerant to egg, and you can see a very high production of interferon gamma and very little production of IL-4, which translates to a higher Th1, Th2 ratio. In other words, saying that the Th1, Th2 ratio is much significantly lower when you're allergic to that particular food product. Interestingly, in kids who have outgrown allergy or who are not allergic to peanut, you'll see that there's an increase in the Th1, Th2 response, highlighting that the Th1, Th2 ratio or the T cell phenotype, which is specific to the allergen, has an important bearing on the allergic status of the individual. And then this brings us to our working hypothesis. We believe that in peanut allergic patients, desensitization and or tolerance to peanut can be achieved by introducing a Th1 bias to the peanut-specific T cell response. That is, we shift the balance or modulate the immune response. Now, how do we bring about this? The way we bring about this, or the way we have gone about to solve this problem, is to introduce those peanut allergens, the peanut antigens, into a viral vector, so that when the body sees it, it's packaged within the virus, it sees the peanut antigen as a viral antigen and induces a Th1 response. Now, the viral vector that we have chosen to go with is the Cementis Copenhagen vector vaccine platform. Now, this is a proprietary replication deficient vaccine platform. This has been proven to be safe and highly immunogenic in infectious disease vaccine models. And the peanut hypoallergenic vaccine is the first of our vaccines looking into immunomodulatory or along the allergy pipeline. Now, what we have done is we have packaged the peanut antigens, both the major and the minor antigens, as polyproteins, which are under a ubiquitin signal. And the reason we have added the ubiquitin signal is to promote the degradation of these peanut antigen, and this in turn then prevents the extracellular secretion of antigens and therefore an allergic reaction. Now, in the bottom panel here, this is just a characterization or the validation of our vaccine, which we do by PCR, Sanger sequencing, as well as next-gen sequencing. In the bottom right-hand panel is a Western blot, and this Western blot confirms our theory behind adding the ubiquitin signal. We could only detect peanut antigens only when we suppress the proteasomal degradation using a compound which is called MG132, and it was only when we suppress the proteasomal degradation, or that is suppress the ubiquitin signals, did we actually see the peanut antigens, suggesting that our theory of adding the ubiquitin signal does enhance the safety profile and will also increase the T cell presentation. Now, with the vaccine now constructed and validated, we now set out to answer a few key questions. We started off with, does the vaccine induce a tolerant peanut-specific immune response? To answer this question, we vaccinated the mice in a prime boost strategy, and then looked at the T cell responses and the B cell responses. We could see that in these vaccinated mice, the T cells, the peanut-specific T cells, were predominantly secreted interferon gamma, whereas in the control sensitized mice, you could see that there was a higher proportion of IL-4 producing T cells, suggesting that the vaccine induces T cells with a higher Th1, Th2 ratio. Correspondingly, we see that there's an increase in the tolerant IgG2 antibody response with little IgG1 and no IgE antibody response. Next, we question, if the vaccine can prevent peanut sensitization or peanut allergy, or in other words, we were testing the prophylactic potential of the vaccine. In order to test this, we vaccinated the mice, and following vaccination and confirmation that there was a Th1 biased T cell response and a tolerant IgG2 antibody being produced, we force sensitized the mice using an Th2 alum adjuvant. And what we do see is that the vaccinated mice maintained Th1 bias or the non-allergic status. And the proof for that is in the bottom panel. And I'll take you through this. So in the vaccinated mice, you'll see that there is a tolerant IgG2 response, whereas in the non-vaccinated mice, we see that there is an IgE antibody response. The Th1, Th2 ratio was significantly higher in the vaccinated mice, as well as the mast cell degranulation was reduced in the vaccinated mice. This tells us that there is a Th1 bias across all the three key cellular players of the allergy axis, the B cells, the T cells, as well as the mast cells. Now, upon feeding these mice with peanut, we saw that it was only in the non-vaccinated mice did we see the lymphoplasmacytic infiltration within the guts, but this was completely not seen in any of the vaccinated mice. This goes on to tell us that this Th1 biasing in the protective efficacy that we see is not only systemic, but it's also at the mucosal surface, at the gut mucosal surface, or the mucosal point of contact with the allergen. Next, we question if the vaccine was safe to administer in an allergic setting. In order to do that, we first sensitized the mice to peanut using, again, the alum adjuvant, which induces a Th2 biased T cell responses, as well as induces IgE antibody responses. And following that, we vaccinated the mice. Following vaccination, we observed the mice over a 12-hour period, and we scored the mice against a clinically approved matrix to grade allergic reactions. We saw no signs of allergic reaction. We had temperature transponders in these mice in order to make sure that there was no drop in temperature, which is one of the signs of anaphylactic or allergic reaction. Now, once we established the vaccine was safe to administer in an allergic setting, our next important question was, can the vaccine modulate the immune responses in these allergic mice? So in order to do that, we sensitized the mice, confirmed the sensitization profile, and then followed that with vaccination in a prime boost setting. And what we saw that even in the sensitized mice, vaccination could bring about an increase in the tolerant IgG2 antibody levels. It brought about an increase in the Th1, Th2 ratio, and it brought about a slight fall in the mast cell degranulation levels. So we then next moved on to see if we could translate this into human cultures. In order to do that, we collected blood from peanut allergic individuals. We isolated dendritic cells, and then we pulsed the dendritic cells with either peanut or with the vaccine, and then put them up into culture with CD4 T cells, or autologous CD4 T cells, and then we maintained them in culture for about 3 weeks. At the end of 3 weeks, we fed peanut back into the cultures, and then we looked at the cytokine profile of the T cells and examined the Th1, Th2 ratio. And you can see here that in the cells which were pulsed with peanut, or the unvaccinated cultures, you could see that there is a higher proportion of IL-4 being produced, whereas in the cells which were pulsed with a vaccine, you could see that there's a significant increase in the amount of interferon gamma production. This in turn resulted in a significant increase in the Th1, Th2 ratio in the cultures. We went ahead, and in the interest of time, I haven't shown you the results, but we were able to show that these assays were all reproducible. The vaccines induced Th1 biased T cell responses, which are antigen specific, and that the antigen specific Th1 bias could be maintained in long-term cultures.
Thank you, Preethi. We might need to finish there. Thanks. So do you have a conclusion slide?
Yeah, so this is the conclusion. So we were hoping that we could, we have just received a NHMRC IDEAS grant, and we are hoping that we can gear up to start testing the protective efficacy, as well as understand the capacity of the vaccine to induce a long-lived tolerant response, both systemically as well as within the gut mucosa. We're very keen to explore antigen-specific T-cell responses, T-regulatory T-cell responses, CD8 T-cell responses. We're keen to understand the transcriptome and the methylome changes which happen during the immunomodulation. So we're quite keen to collaborate to help answer these questions and get a better understanding. And I'll leave you guys with the acknowledgement site, the key players at the Experimental Therapeutic Lab. Dr. William has been involved in the project right from the conception, and Adriana has helped us with the blood sample collection for all the human samples. Dr. Jimmy will help us with all the bioinformatic analysis for the transcriptome and methylome. And Dr. Andreas at JCU will help us refine the model in terms of gut microbiome.
Thank you. Thanks very much, Preethi. I'm sorry we do need to move on, but thanks very much for that. I do see there's no questions in the Q&A chat, and I think probably in the interest of time we will move on, but thanks very much for that presentation.
Thank you.
I'd like to introduce our next speakers now, if I may. Can we change slides, please? So next we have from the Royal Adelaide Hospital, Clinical Immunology and Allergy Division. The speakers are Dr. Adriana Lee and Dr. Pravin Hissaria, senior consultants with immunology and allergy clinicians. And I'd like to also acknowledge Professor Bob Heddle in developing this project. So without further ado, please, Adriana and Pravin, go ahead with this talk. Thanks.
So I'd like to thank John for giving us this opportunity to present our research on Jack-Jumper allergy, of which Professor Heddle started this research. Just an overview of the presentation today, so some background on the problem, the initial studies of Jack-Jumper ant venom immunotherapy, and our preliminary data to date. So just to give you some background, Jack-Jumper ants, the scientific name is Myrmecia pilosula. They're also known as hopper ants in South Australia, jumping jack ants in other parts of Australia. They're only just over a centimetre, but they really pack a punch. They're actually the second most common cause of insect anaphylaxis after bee sting in South Australia. And they are a dominant cause of insect anaphylaxis, and the predominant cause in Tasmania, but also a very important cause of insect anaphylaxis in regional areas of New South Wales and Victoria. There have been certainly fatalities associated with anaphylaxis to hopper ant stings prior to the advent of immunotherapy. There are at least 5 deaths in Tasmania and probably a couple of deaths in South Australia. So that's why it's an important problem. So without immunotherapy, the management of insect allergy is trying to avoid the stings, which is often not possible, or carrying an EpiPen, which has several limitations, one of which is that sometimes there's not an adequate dose of adrenaline to treat severe reactions. It's common to forget to carry your EpiPen. And carrying an EpiPen actually doesn't improve your quality of life because there's a high degree of anxiety and overvigilance associated once you have a fear of anaphylaxis with stings to that culprit insect. And so that's why disease-modifying treatment like venom immunotherapy or desensitization is a key management strategy. So the principles of all venom immunotherapy or other allergen immunotherapy, such as even peanut immunotherapy that Preethi was talking about, is to induce tolerance. The strategy for that is to start off with a very small dose of the allergen, in this case the Jack-Jumper ant venom, and to increase the dose gradually until you reach a target maintenance dose, at which point you will have hopefully induced clinical tolerance, and then to administer that maintenance dose regularly over a prolonged period of 3 to 5 years, such that you induce long-term immunological tolerance, such that even when you stop treatment, hopefully the patient will retain that clinical tolerance. The mechanisms of venom immunotherapy and allergen immunotherapy in general are not sort of generally but not completely understood. They're thought to induce allergen-specific T regulatory cells and alter the balance in favour of the production of allergen-specific T regulatory and Th1 cells and skew away from Th2, allergen-specific Th2 cell production, which then the production of T regulatory cells is associated with the production of regulatory cytokines like interleukin-10 and TGF-beta, which then induces the production of B cells, which produce protective allergen-specific antibodies such as IgG4 and IgA, and the suppression of allergen-specific IgE, as well as the suppression of effector allergy cells such as mast cells and basophils. So this led to a landmark trial of Professor Hedl and Professor Brown's in the early 2000s, where they did a landmark trial of Jack-Jumperant venom immunotherapy. This was actually only the third ever randomized trial of venom immunotherapy in the world. The first two before that were in the 1970s to be venom treatments that hadn't been done in a long time. They recruited 68 healthy adult volunteers in Tasmania with a history of anaphylaxis to Jack-Jumperant. And they allocated them to receive either placebo or 100 microgram maintenance dose of Jack-Jumperant venom. And these brave individuals then subjected themselves to a live ant sting challenge in the Royal Adelaide Hospital Emergency Department, which I have a video of what the sting challenge looks like. Hopefully it plays. Oh, well, QuickTime's not available. Sorry. So in this trial, 1/3 of patients had some form of systemic allergic reaction to the treatment itself, so those injections. But the majority of those reactions were mild, only 2 were associated with hypotension. Then in the initial double-blind phase of the trial, the patients were subjected to the live ant sting challenge. So these patients are stung once, and then if they don't react after 45 minutes, they're stung the second time in case the ant sac was empty. And in the case of the initial double-blind phase trial, 72% of subjects who received placebo immunotherapy had a generalized allergic reaction, 8 of which were severe hypotension. And none of the patients who were in the Jack-Jumperant active treatment arm had anaphylaxis. And because of this interim analysis at that point, it was decided to switch over all of the patients in the placebo arm to active treatment. And then all these patients went on to have stings as well. And only 2 patients had a reaction to the sting challenge, and they were very mild. They were just generalized skin symptoms where they previously had a history of anaphylaxis. It was shown to be very effective treatment. So 100 micrograms of venom as treatment was found to be very effective. That said, Professor Hedl then went and Dr. Brown then went on to further studies to try and refine the treatment more. They compared build-up schedules of weekly build-up over around 10 weeks versus a very rapid build-up to the top dose over a couple of weeks. And they found that, unfortunately, the rapid build-up was much less well tolerated, with nearly 2/3 of patients having systemic reaction versus 1/3 with the slower build-up. And then they also compared a top maintenance dose of 100 micrograms versus 50 micrograms and found that 50 micrograms was just as effective and better tolerated. So for this reason, the standard of treatment of venom, Jack-Jumperant venom immunotherapy, where in areas where it is available in Australia, is to do a slow build-up over 10 weeks and then to give to a target dose of 50 micrograms and then to give that standard dose over a prolonged period for 5 years. Now, while it's a very effective treatment, there's certain limitations to it. One major limitation is that this venom is not cheap and it's not easy to collect. It literally involves people driving up to the hills in Tasmania, vacuuming up the ants, freezing them, and then individually dissecting the ant sacs under microscopy to dissect the venom. So this means that the venom is for patient around $3,000 per year, and these patients are having 5 years of treatment, so the cost does add up. It's a prolonged course. A significant minority of patients aren't able to achieve maintenance doses due to side effects. If we could have a treatment that would allow us to use less venom and to have less side effects, or to achieve effective treatment in those who weren't able to achieve it with standard dosing, then obviously that would be great. So one way to do that is to use adjuvants. So as we know, adjuvants are substances that are co-administered with vaccines, or in this case, an allergen extract, to enhance the desired immune response. In this case, the desired immune response is to enhance T regulatory and Th1 and IgG responses, and to suppress Th2 and IgE responses. And there's numerous adjuvants available, alum being the traditional one, but another delivery system that's novel is delta inulin, which is the one that was used in our current trial, and also TLR agonists such as CPG and MPLA4. Delta inulin was developed by Professor Nick Podrowski with his company Vaccine, based in Flinders, with the NIH adjuvant development grant. It is a naturally occurring plant-based polysaccharide, which has been shown in previous trials, not just for bee venom treatment, but also for influenza and hepatitis B, and most recently it's been used in even a COVID vaccine. But it's been shown to promote a strong Th1 response without producing pro-inflammatory cytokine effects, and to skew away from a Th2 response, which is desirable. And so that was the basis for using it in our study. In the bee venom trial, it had been shown to produce robust IgG4 responses. That said, that bee venom trial of Professor Heddles and Dr. Hissaria's was limited by a lack of formal sting challenge at the end. There's also a possibility for a future trial where we'll combine Advax with CPG, which is stimulates TLR9, and which then produces interferon gamma and interleukin 12, which will then skew away from a Th2 response, which would also be desirable. So this led to our JumpVax trial, which is our current trial, which is a investigator-initiated trial where subjects recruited to both Royal Adelaide and Royal Hobart Hospital to receive either 50 or 25 micrograms of maintenance dose of Jack Jumper ant venom with or without the delta inulin. After 12 months of these maintenance injections, we then do a live sting challenge of the patients with ants freshly caught from the Adelaide Hills. We also received funding for a supplementary study to look at basal activation testing concurrently, to see whether that can be used as a biomarker to predict effectiveness of treatment and side effects of treatment. This is our preliminary clinical data. We screened 49 patients between Royal Adelaide and Royal Hobart. 5 didn't proceed because they were ineligible or they elected not to proceed. 44 were randomized to commence treatment. Of these patients, 1 subject withdrew early in build-up due to reasons unrelated to the trial. 1 subject withdrew near the end of treatment due to fears of attending hospital with the COVID pandemic, but then did consent, was continued out of trial on the same dose, and then actually subsequently consented to an unblinded sting challenge. 2 subjects withdrew actually due to recurrent systemic allergic reactions, and were unable to consistently tolerate the target maintenance dose, so would be considered treatment failures. And 4 patients are still on active treatment awaiting their sting challenge. So that means 36 though did complete 12 months of maintenance treatment as planned, and all proceeded to sting challenges. And of the 37 who did complete treatment or near complete treatment and then proceeded to sting challenge, 84% had a negative sting challenge. So that means 16% did have a positive sting challenge. That said, none of them were severe or were grade 1, which just means hives or itch without anaphylaxis, whereas the majority of our patients had initially had a history of anaphylaxis. So it can be concluded actually that of all the patients who managed to reach maintenance and tolerate maintenance, they received some degree of protection and all were protected against anaphylaxis. That said, around half of our patients did have some form of systemic reaction during treatment. The majority of these reactions, however, were mild grade 1 reactions and were self-limited to 1 or 2 reactions. That said, 6 subjects had 3 or more systemic reactions, of which 4 of them were classified as grade 2 or anaphylactic type systemic reactions. With that said, I'll pass over to my colleague, Dr. Hissaria, to present the preliminary laboratory data.
Yeah, hello everyone, and sorry to go on a bit far in the time, so I'll try to keep it very short. So as Professor Hedel said about designing this trial and doing such extensive thing, what could be seen was this was a unique opportunity for us to do some lab work to make sense of some cellular or biochemical events that might be happening, which could give us some insight into the various regulatory mechanisms that might underlie the tolerance induction. We had basophil activation test. I won't go into details of this test. I'm sure you must have heard it before, but it is a very simple flow cytometry based test, which we have now optimized in our lab, whereby you can take the whole blood from the participants. We can put in the allergen to which they are allergic to, and then we look at activation of the basophils by looking at the upregulation of CD63. So we decided to use that as part of this clinical trial to see if that could be used as a biomarker. We already had, Professor Hedel already had developed specific IgE to JAK jumper and Venom, and we used that same platform to actually design for IgG4 analysis as well. And that is because the specific IgE to JAK jumperant is by no means optimal. It misses about 15% of cases. And as you can see here, we found that basophil activation test was as good as Venom skin test, which is not routinely available. Just a few slides about what we have found so far. So one is we found that there was marked reduction in the basophil activation after the immunotherapy. As you can see here, the A is with 0.1 microgram per mil of the Venom, and the B is 0.01. And as you can see, there was marked reduction in the percentage of basophils which were activated, although it was still present at the higher concentration of 0.1. Subsequently, as I said, we also were looking for specific IgG4 tests, which we had standardized by then as well. And as you can see nicely here, although IgE, the goal is to reduction of IgE in long term, as you can see during immunotherapy anyways, there is actual increase in the IgE to Venom that was found after first maintenance, and subsequently, even at the 12-month mark, it was higher than what could be screened at the screening level. However, what we really got excited was that there was a definite signal towards an increase in the IgG4, which could be seen as early as first maintenance. So this is within 4 weeks of commencing their immunotherapy, and which then subsequently gradually increased as the time passed to a maximum at 12 months. And that is what we were trying to show here, that although the changes in the IgE levels at 0 and 12 months were not significant, there was an increase in the IgG4 levels, and the IgE by IgG4 ratio was significantly reduced as well. So there must be something that the IgG4 was doing. And this is not a new thing, to be honest. This has been documented in other models of allergies as well. But that is what we are trying to now look forward as to what this G4 actually might be doing in terms of the immunological mechanisms. Again, this is just a negative and positive challenge of our patients who have completed the 12 months so far. And as you can see, there was a very high negative predictive value as to most of the people who had a negative challenge had marked reduction in their basophil activation. As you can see here, apart from these 2 outliers. The positive predictive value was not quite high because there were some negative challenges as well, but definitely negative. So if you had basophil activation tests which had significantly reduced, they might be the ones who are becoming more tolerant. So look at this forward now, we have put up a subsequent grant, which is basically looking at just the mechanisms of IgG4 and the basophil reactivity reducing. And we think that this increase in specific IgE4 is not just mopping up the venom as the venom is introduced after a sting, but it actually might be causing some immunological changes by causing reduction in the in the uptake of allergen by IgE mediated mechanisms. And that the increasing levels of G4 and the falling of the basophil reactivity might be correlating with the degree of desensitization and might be used as a marker to decide on the duration of the immunotherapy. And that the combination of these tests will be what is most important. And our research plan currently is that we are trying to set up this IgE-PHAV assay, which is basically an B-cell mediated assay, whereby you look at the mast cell line, and then you look at the allergen uptake and whether it is blocked by pre-incubating it with the IgG4. We are looking at the Western blots, and that is to look at specificity of this G4, because just generalized increase might not be important. It might be the increase of IgG4 to a specific allergen, and the similar way mast cell degranulation assay. I guess the last slide is basically, I think part of this seminar is to initiate collaborations. And there is very little immunology that has been done, at least when we started this project way back in 2018. Last 2 years, we've seen significant progress with some human immunology work as well. There has always been a good animal allergy work. But this is a very good human model of allergy, and we have different cells and sera stored at different time points, which could be used to test for various immunological mediators, whether be it using proteomics to look for soluble biomarkers, or to look at microbiome along with it, or to look at some genetic biomarkers, because we have stored the mRNA at different stages as well. And I think I'll stop at that, and with a photo of Professor Hedel sitting on an anthill.
Thank you very much, Pravin and Adriana and Preethi. Thank you for those excellent 8-minute talks. Very good. I'd like to wind up now and apologise for going on a little bit over time. I see again there are no open questions, and look, in the interest of time, I think we'll have to bypass questions, but I'd like to thank the presenters for their excellent talks today, showing that there is indeed some immunology in allergy at last on both sides. So thank you very much for that once again. Next Tuesday's focus will be on endocrinology with Professor Michael Horowitz and Professor Gary Whittett and Andrew Zanatino as chair. So thank you very much for your attention and your attendance, and see you next week.